I welcome all the registered students in SWAYAM course of genetic engineering and its recent advancement. In previous lectures, you have been gone through the topic genetic manipulation of cells. The topic that we will cover in today's lecture is types of transfections. I am Dr. Kush Kumar Nayak, Assistant Professor, Amity Institute of Biotechnology, Amity University, Chhattisgarh. Today I will cover the following content in present lecture. We will start with introduction, then we will move to understand what is transfection. We will check overview of transfection techniques. Under this, we will see chemical transfection method, which consider calcium phosphate transfection, lipid based transfection, polyethylenamine transfection, detergent transfection, DEA dextran based transfection, and then we will move into the next part related to viral transfection method, which cover adenovirus, retrovirus, lentivirus transfections. Then we also see the physical mode of transfection method, this having electroporation, microinjection, particle bombardment, sonoporation and magnetofections transfection methods. After that, we will see the various application of transfection techniques. So, let us start with the introductory part of the transfections. So, transfection is a technique used in molecular biology and biotechnology to introduce foreign nucleic acids such as a DNA or RNA into the cell. It enables researchers to manipulate and study genes, protein expression and cellular functions. By delivering exogenous genetic material into the cell, transfection allow for investigation of gene function, protein production and development of therapeutics for various diseases. Transfection can be achieved through different methods, which can be broadly categorized in three categories. First, chemical transfection mode, second viral transfection and third one is physical transfection. For example, electroporation and magnetofection, which come under physical methods. We will see the details one by one in upcoming slides. So, let us first understand why transfection is important, why there is a need of transfection. So, transfection play a crucial role in molecular biology and biotechnology, providing significant advantages and contributing to advancement in various research areas. Here are the certain key significance of transfection which we should be keep in our mind. First, related to gene function in studies, transfection allow researchers to introduce exogenous DNA, RNA into the cells, enabling the study of gene function. By manipulation gene expression, researchers can investigate the role of specific gene in cellular processes, development and disease. Transfection facilitates the expression of a production of a specific protein of interest in cell. This is particularly valuable for producing recombinant protein for therapeutic purpose, biotechnological application and studying protein function and interactions. Another area where it used is disease research and drug delivery, also related to the drug discovery. Transfection helps researchers understand the molecular mechanism underlying disease. By introducing disease related genes or specific mutation into cell or model organism, researchers can study disease progression, identify potential drug target and test the efficiency of therapeutic interventions. Another key point is genetic engineering and biotechnology, where transfection allow for the introduction of a desired genes or DNA fragment into the cells, enabling the production of transgenic organism, creation of a genetically modified cell lines and development of a genetically engineered crops or animals for various purposes. Another key area where it uses therapeutic application, where transfection play critical role in a gene therapy, a promising approach for treating genetic disorder and certain type of cancer. By delivering therapeutic genes into target cells, transfection can be used to correct genetic mutation, restore normal gene functions or modulate gene expression, with the potential to provide long term therapeutic benefits. Again, a functional genomics and high throughput screening is the area where it also have an important application. Transfection techniques widely utilized in functional genomics studies and high throughput screening assay. By introducing siRNA, shRNA or CRISPR-Cas9 based gene editing tool, researchers can systematically silence or modify specific gene, enabling the identification and characterization of gene functions. Another key area where it used is cell line development. Transfection is crucial in creation of a stable cell line expressing specific genes of interest. These engineered cell lines serve as a valuable tool for studying gene function, drug screening, protein production, and biopharmaceutical developments. So, overall, transfection techniques are powerful tools that enable researchers to manipulate and study the genes, proteins, cellular process, paving the way for advances in molecular biology, biotechnology, and in therapeutic research area. So, let us discuss about the various techniques that is used for transfection purpose. The techniques that facilitate the uptake of nucleic acid into the cells are categorized in three common way, as we said it: chemical transfection, viral transfection, and the physical transfection. In chemical transfection method, the calcium phosphate transfection, lipid based transfection, dextran transfection are the common method which apply. Chemical transfection method are relatively simple, cost effective, widely applicable to different cell types. However, they can exhibit lower transfection efficiency and may induce cytotoxic or inflammatory response into the cells. On the other hand, viral transfection method utilize viral vectors or modified viruses to introduce nucleic acid into the target cell. The most common viral vector include adenoviruses, can accommodate large DNA insert efficiently, in fact both dividing non-dividing cells. They typically do not integrate into the host genome, but provide transient transgene expression. Another virus is class of retrovirus, such as lentivirus utilize a reverse transcriptase process to integrate their genetic material into the host genome. This enables stable and long term transgene expression. Another category of virus is adeno associated virus, that is also known as AAV. These AAV vector have a low immunogenicity and can enable long term transgene expression without integrating into the host genome. They are useful for certain gene therapy applications. Viral transfection method often yield high transfection efficiency, but they require specialized viral production and safety precautions. The use of viral vector may also raise concern regarding immunogenicity and potential insertional mutagenesis. The third category of transfection is counted as a physical transfection. These are the technique rely on physical forces to deliver nucleic acid into the cells. These methods include like electroporations, in which cells are exposed to brief electric pulses, creating temporary pores in the cell's membrane that allow DNA to enter. Another technique is magnetofaction, in which magnetic nanoparticles are complexed with DNA and directed to target cells using a magnetic field. Another example is sonoporation, which pulse ultrasound waves create transient membrane disruption, enable the uptake of DNA. So, physical transfection method offer high transfection efficiency and can be used a wide range of cell types. However, they may include cellular damage or require specialized equipment. So, each transfection techniques has its advantage, limitations and optimal applications. The choice of a specific method depend on factors such as a cell type, desired transient expression duration, transfection efficiency, cytotoxicity and the purpose of the study. Researchers often need to optimize transfection condition to achieve optimal result for their specific experimental needs. So, let us discuss each transfection types in detail one by one. The first method of transfection, that is known as a chemical transfection method. Such method are laboratory technique used to introduce foreign genetic material such as a DNA or RNA inside the living cells. This method involve uses of various chemical reagent to create temporary pore or opening on the cell membrane, allowing the entry of foreign genetic material into the cell. This enables the manipulation and study of cells, genetic makeup, cell gene expressions and protein production. There are several types of transfection method that are come under chemical transfection, out of which the predominant and most popular method is known as a calcium phosphate transfection method, in which calcium chloride and phosphate are mixed with DNA to form a calcium phosphate precipitate. The precipitate adhere to the cell surface and is taken up by cell through endocytosis. Once inside the cell, DNA is released and transported into the nucleus. Another mode of transfer of a DNA material in the cell is liposome mediated transfection, where liposomes, which is a lipid based vesicles, can fuse with the cell membranes, delivering genetic material into the cells. Liposomes can be positively charged or neutral molecule. Cationic liposomes form complex with a DNA, which are then taken up by cell through endocytosis. The complex are subsequently released into the cytoplasm and transported into the nucleus. Another chemical through which transfection done is polyethylene amine, also known as a PEI. This PEI is a positively charged polymer that can condense and protect the DNA, allowing the entry into the cell. PEI does not require the use of transfection reagent and can be directly mixed with the DNA. Once inside the cell, PEI aid in the release of DNA from endosomes, allowing it to reach the nucleus. Detergent also used for transfection purpose. In this detergent like digitonin or triton X100 to create pores in the cell's membrane, allowing the entry of a genetic material. The detergent form complex with the DNA, which can then press through the pores into the cells. Overall, chemical transfection method provide a versatile and widely applicable approaches for introducing foreign genetic material into the cell, enabling genetic engineering, gene therapy and various researches in molecular biology. Let us understand the process involved in this different method. The first method that is known as a calcium phosphate transfection method, the steps are as follow. Here the calcium chloride and phosphate combine with a DNA of interest, form a calcium phosphate precipitate, adhere to the cell surface, which is then taken up by cells through endocytosis. Once it goes inside the cells, DNA is released and transported into the nucleus. The detailed process are started with the preparation of a DNA calcium phosphate complex. The DNA of interest is typically prepared in a separate solution and mixed with an equal volume of 2x HbS buffer. The calcium chloride concentration is critical for efficient transfection and optimal condition may require optimizations. In line of this, formation of a calcium phosphate precipitate is required. To form precipitate, DNA calcium mixture is vortexed or gently mixed and incubated at a room temperature for a specific period of a time. During this time, calcium phosphate precipitates form from the combination of a DNA and calcium chloride. Afterward, precipitate should be introduced inside the cells. The DNA calcium phosphate precipitate is added directly into the cells in culture medium. The concentration of a DNA in transfection mixture should be optimized for each specific cell type and applications, as too much DNA can be toxic and reduce transfection efficiency. Afterwards, the step follow is a cell incubation, in which after the addition of a precipitate, cell are typically incubated at 37 degrees centigrade in CO2 incubator for several hours. During this time, the precipitate adhere to the cell surface and is taken up via endocytosis. Afterward, release of DNA and nuclear transport must be taken care about. Once inside the cell, the DNA is released from the precipitate and likely facilitated by the low pH environment of the endosomes. After that, the another category or another example of a chemical based transfection method is we can count or we can say is a lipid based transfection method. Here liposome mediated transfection is widely used for introducing genetic material into the cells. Liposomes are a lipid based vesicle that can fuse with the cell's membrane, allowing the delivery of a DNA or RNA into cell. The steps involving in this method started with the liposome preparation, where liposomes used to transfection are typically made from a cationic lipids. Cationic lipids have a positive charge that helps them interact with the negatively charged nucleic acid. The lipids are dissolved in an organic solvent such as a chloroform or ethanol and then dried under a vacuum to form a thin lipid film. Afterward, DNA complex formation have been taken care about. For that, the lipid film is dehydrated with a solution containing the genetic materials of interest These lead to the formation of a liposome DNA complex, where the genetic material become encapsulated or associated with the liposome. In the third step of this methods, incubation of liposome DNA complex is done with cell. The liposome's DNA complex are added to the target cell in a culture medium. The concentration of a complex and duration incubation can vary depending on the specific cell line and transfection efficiency required. After that, fusion of a liposomes with a cell membrane is taken care about. The cationic liposomes interact with the negatively charged cell membrane through electrostatic interaction. This leads to the fusion of the liposome with the cell membrane, allowing the liposome's DNA complex to enter the cell. After this step, the next is followed by endocytosis and endosomal escape. Once the liposome DNA complex are taken up by cell, they are often internalized through a process called endocytosis. The complex are then trapped within the endosomes, which are membrane bound vesicles with the cells. Efficient transfection require the complex to escape from the endosomes into the cytoplasm, for which several mechanisms can facilitate release of DNA from the endosome into the cytoplasm. One possible mechanism involves the pH sensitivity of a liposomes. The acidic pH in endosome trigger a change in a liposomes causes destabilization and subsequent release of the DNA payload into the cytoplasm. Once the DNA released into the cytoplasm, it should be transported into the nucleus. So, release of DNA must reach to the nucleus of the cells to exert its effect. The DNA can enter the nucleus through nuclear membrane pores or with the help of nuclear targeting signal present in the DNA sequence. The exact mechanism of the nuclear import may vary depending on the cell type and the characteristics of the DNA. The last, gene expression and analysis, where once the DNA come inside the nucleus, the transferred DNA can integrate it into the host genome and exit temporarily as an episomes. The genetic material can then be transcribed into RNA and translated into the proteins, allowing for the expression of introduced gene or other genetic manipulation. It is important to note that optimization of a liposome composition ratio, cell culture condition are necessary for maximizing transfection efficiency, minimizing cytotoxicity and obtaining reliable experimental results. Additionally, transfection efficiency and efficacy of a liposome mediated transfection can vary among different cell types and must be carefully considered when designing an experiment. The next chemical that we used under chemical based transfection method is a polyethylenamine. Transfection is another commonly used method for the introducing genetic material into the cells. The PEI is a positively charged polymer that can condense the product DNA that can condense and protect DNA, facilitating its entry into the cells. The steps involved in PE based transfection methods started with the preparation of a DNA PE complex. DNA of interest is prepared in a separate solution at an appropriate concentration, and PE is then added to the DNA solution at a specific ratio to form complex. The ratio of PE to DNA can vary depending on the cell type, and application and optimization may be required for this step. The complex formation is then observed in next step. The mixture of a DNA and PE is gently mixed and incubated for a certain period of time to allow formation of a complex, a stable complex. The PE bind to DNA molecule, neutralizing their negative charge and forming compact structure that protect DNA from degradation. Afterwards, addition of complex to the cells is a crucial step, where DNA PE complex are added directly to the cells in a culture medium. The concentration of a complex should be optimized based on a specific requirement of a cell type and application. Care should be taken to avoid excessive complex concentration, as high concentration of PI can be toxic to the cells. Once it is transferred successfully inside the cells, the next step that relates it about its incubation. After the addition of a DNA PI complex, cells typically incubated at 37 degrees centigrade in CO2 incubator for a specific period of a time. The incubation duration may vary, but it generally is sufficient to allow the PI complex to interact with the cell membrane and enter into the cell. Further, intracellular delivery, the PI DNA complex are though to enter cells primarily through endocytosis. The complexes are engulfed by the cell membrane and enclosed in a vesicle called endosomes. Afterwards, it should escape from the endosomal area and enter into the cytoplasmic area. The transfection required DNA to escape from endosomes and reach to the cytoplasm, where it can access the cellular machinery for gene expression. The PI has been shown to induce swelling and rupture of endosomes, promoting the release of DNA into the cytoplasm. The exact mechanism of this activity is not fully understood yet, though it can vary depend on the specific PI formulation and the cell type. And the last, once DNA PI complex come into the cytoplasm, it must be go to the nucleus. The once cytoplasmic PI DNA complex is available, it should be reached to the nucleus for a gene expression, and the DNA can enter the nucleus through nuclear membrane pore or with the help of nuclear targeting signal present in the DNA sequence. Afterwards, gene expression and analysis is going on by the help of transcription translation machinery of the cells. The genetic material can then be transcribed into RNA and translated into the protein, enabling the expression of the introduced gene or genetic materials. So, it is important to optimize the PI DNA ratio, complex concentration and culture condition to achieve efficient transfection and minimize potential cytotoxicity. The another chemical method is utilization of a detergent, where detergent is being used to introduce genetic material into the cell by creating pore or openings directly into the cell membrane. Detergents disrupt the lipid bilayer structure of cell membrane, allowing the entry of foreign genetic material. First, we have to select detergent very carefully for start this process. Different detergent can be used for transfection, such as a digitonin or Triton X100. The choice of certain detergent depend on the specific cell type, transfection requirement. So, digitonin is a mild detergent that form transient pores in a cell membrane, allowing DNA or RNA to enter in the cells, while Triton X100 is more potent and can permeabilize the cell membrane more extensively. So, preparation of a DNA and RNA for such activity, genetic material must be prepared in a separate solution at an appropriate concentration, which will be then mixed with the detergent to form complex. Detergent can help associate the genetic material with lipid based carriers such as liposomes or micelles, which aid in protecting the nucleic acid and facilitate their delivery. Addition of a complex to cells, the DNA or RNA detergent complex are added directly to the cells in a culture, the concentration of which are optimized based on cell type and desired transfection efficiency. At the last, cell must be incubated at particular temperature for a particular period of a time, an appropriate temperature which can vary depending on the cell type. During this incubation period, the detergent disrupts the cell membrane, creating temporary pore that allow the entry of a genetic material into the cells, after which DNA release and it will be uptake by the nucleus. The detergent help in the release to the genetic material from the complexes, making it available for uptake by the cells. DNA or RNA can then enter the cell through the pores in the cell membrane. After that, in the next step, which one is a crucial part, where endosomal escape and nuclear import must be happen with neat and clean process. The genetic material may need to escape from endosomes to enter into nucleus to exert its effect. These processes can vary depending on the cell type and nature of genetic material being transfected. Detergent transfection can be efficient method for introducing genetic material into certain cell types. However, it is essential to carefully optimize the concentration of a detergent and incubation time to avoid excessive cell damage or cytotoxicity. Additionally, it is important to note that some detergent may disrupt cellular process or induce cell responses which can affect the interpretation of experimental result. Another category under this method is a DEAE dextran transfection. So, DEAE dextran transfection is commonly used method for introducing nucleic acid into mammal cell, where DEAE dextran, which stand for actually dimethyl aminoethyl dextran, which is a cationic polymer that can facilitate the uptake of a nucleic acid by the cell. The steps involved in this process is we start with the preparation of a nucleic acid solution. DNA or RNA to be transfected is prepared in a separate solution at an appropriate concentration. The nucleic acid then isolated from a plasmid PCR product or synthesized from a template. After that, DEAE dextran is added into this nucleic acid solution, usually at a specific ratio to form complex. DEAE dextran bind to the nucleic acid, protecting them from degradation and facilitate cellular uptake. The DNA or RNA that complex with the DAE are added directly into the cells in culture medium. The concentration of complex should be optimized based on cell type and desired transfection efficiency. Afterwards, cell must be incubated. Cells are incubated at an appropriate temperature, usually near 37 degrees centigrade, for a specific duration to allow complex to interact with cell membrane and entered inside the cell. Afterwards, endocytosis and escape from the endosomes is happen in the similar way as we discussed in previous slides, through which it infuse inside the nucleus of a cell. The efficient transfection require the complex to escape from the endosomes into the cytoplasm. So, entry into the nucleus of this DNA that bind with the DAE dextrose complex is taken care by the cellular mechanism itself. The nucleic acid can enter into nucleus through nuclear membrane pore or with the help of nuclear target signal present in a target gene or target DNA sequence. DEA dextran transfection is generally considered as a simple and cost effective method for introducing nucleic acid into mammalian cells. However, its efficiency can vary among different cell types. Optimization of a DEA dextran concentration, complex formation condition, culture condition is crucial to achieve optimal transfection efficiency and minimizing cellular toxicity. So, now move towards the second part of transfection method, second category, that is known as a viral mediated transfection method. In this method virus are used as vector to deliver genetic material into the cells. Virus possess unique capability to enter target cells and deliver their genetic cargo. The some key points about viral transfection methods are mentioned below, which is about selection of a viral vector. Various types of viruses can be used as a vector, including retrovirus, lentivirus, adenovirus, adeno-associated virus, herpes simplex virus, etc. The choice of viral vector depend on the factors such as cell type, duration of a gene expression, payload capacity, safety consideration, etc. Another crucial point is construction of a viral vector. The vector is modified to remove or disable gene responsible for the replication and pathogenicity of the virus. The vector is then engineered to carry and express the gene of interest, which is followed by the viral transfection. Means the viral vector carrying the desired genetic material is introduced into the target cell. This can be done by direct application of the vector to the cells through viral particle infection or by packaging the vector into the viral particles. Another point is related to cell entry and transgene expression. Viral vector attached to a specific receptor on the cell surface, enabling the virus to enter the cell. Once inside, the viral genetic material is released and enters into the cell nucleus. The transgene is then integrated into the host genome and exists temporarily as soon as episome. The transgene is transcribed and translated by the cellular machinery, leading to the expression of a gene interest. Advantage of such viral transfection is actually have a lot of point through which we can identify its advantages. Transfection method have several advantages, including high transduction efficiency, long-term gene expression, and the ability to target specific cell types. Viral vector can be engineered to carry large DNA fragment and can infect both dividing and non-dividing cells as well. The safety consideration concern, it is a crucial aspect of a viral transfection method. Care must be taken to ensure that viral vector are replication deficient and do not pose a risk of causing disease. Additionally, the use of appropriate biosafety measures and the consideration of potential immunogenic responses are important consideration for in vivo applications. So, let us discuss certain application of this viral vector method. Viral transfection method have found extensive application in a gene therapy, where they can be used to introduce therapeutic genes into target tissues to treat genetic disorder or other diseases. Viral transfection also play a significant role in research, as it allow for the study of gene function and genetic manipulation in a controlled manner. It is important to note that viral transfection method require careful optimization and validation for each specific cell type and experimental designs. Ethical and safety consideration must also be taken into the account when using viral transfection method for research or therapeutic purpose. Let us understand the use of several virus vectors for such applications. The first example is adenovirus transfection. The different steps of adenovirus transfection typically involve the preparation of a cell, culture of cells of interest is inappropriate growth medium until they reach desired confluence, after which optimize transfection condition. In this, determine the appropriate adenovirus titer and optimize transfection conditions such as the ratio of virus particle to cell as well as the transfection reagent concentration. And the third step, prepare the transfection mixture, where we dilute the adenovirus particle into serum free medium or buffer, add a transfection reagent to the dilute adenovirus suspension and mix gently, and then remove growth medium from the cultured cell using the sterile techniques. Afterwards, add the transfection mixture directly into the cells in culture medium and gently mix to ensure even distributions, through which afterwards the plate has to be incubated in a particular temperature. The cell at 37 degrees centigrade and humidified conditions is kept in 5% CO2 concentration for a defined period of a time, typically between 2 to 24 hours. And after this time, replace the transfection mixture with fresh growth medium. Appropriate supplements such as a serum and antibiotics after the desired transfection method. This supplements must be replaced in a particular after a particular time period. Once we change the new medium into the cell culture, we have to give them time to allow expression and transfected gene. Allow the cells to express the transfected gene for the desired period of a time, which is usually 24 to 72 hours, to observe the effect of the transfection. It is important to note that specific protocol may vary depending on the cell types, nature of adenovirus used, experimental requirement. Therefore, it is critical to consult the literature of a manufacturer guidelines for detailed instruction on adenovirus transfection procedures. Another virus which used in this technique is retrovirus. Retrovirus transfection is a technique commonly used for molecular biology to introduce a foreign genetic material into the cells. Retrovirus are a type of RNA virus that have ability to convert RNA genome into DNA copy through the process of reverse transcription. This DNA copy, known as a provirus, can then integrate into the cell of a host and be stably maintained and expressed. The steps involved in the retrovirus technique is preparation of a packaging cells. Packaging cells that produce the retroviral particle are typically used. These cells are engineered and expressed all necessary viral proteins, but lack the viral genome. Common packaging cells line include HEK293T and Phoenix cells, after which transfected packaging cells. The packaging cells are then transfected with the plasmid vector that combines the gene of interest flanked by the retroviral long terminal repeats. generally known as LTRs. These LTR provide necessary signals for reverse transcription and integration. The transfection is typically achieved using calcium phosphate, liposuction or electroporation technique that allow retrovirus production. After this transfection, packaging cells start reproducing retroviral particles. The particles bud off from the packaging cells and can be collected from the culture medium, which is be then used to concentrate and purify retroviral particles. The collected culture medium is concentrated using techniques such as ultracentrifugation or filtration. The concentrated retroviral particles are then purified to remove contaminants and ensure the high titer functional virus. After this step of infect target cell with the retrovirus, it must be go to allow the transduction and expression of the transgene into the system, which will be then take care by the select or sort transduced cells and ultimately characterize and analyze the transduced cells in the system. Another example of virus mediated transfection is use of lentivirus. Lentivirus transfection is a widely used technique in molecular biology. Gene therapy allow the introduction of foreign genetic material into cell using lentivirus vector. Lentivirus belongs to the retrovirus family and are unique due to their ability to infect both dividing and non-dividing cells. The key steps involved in the lentivirus transfections are actually construct the lentivirus vector, generate lentivirus particles, to collect viral supernatant, then move to concentrate and purify the lentivirus particles, transduce target cells, allow transduction and expression of the transgenes, and ultimately select or enrich transduced cell lines. The third category of transfection is come under physical transfection method. Physical transfection method refers technique that physically disrupt the cell's membrane or provide mechanical forces to facilitate the delivery of a foreign genetic material into the cells. These methods commonly used in molecular biology and genetic engineering experiments to introduce DNA, RNA or other molecules into target cells. Some commonly used physical transfection method are electroporation, microinjection, particle bombardment method, sonoporation, magnetofection, etc. The physical transfection method offer advantage such as a high efficiency, versatility and ability to transfect difficult to transfect cell types. Let us understand all these techniques one by one. In electroporation is a physical transfection method that uses electric pulses create temporary pores in cell membrane allowing the entry of genetic materials. Cells are mixed with desired DNA and RNA molecules and electroporation buffer and subject to brief electric pulses using specialized instrument. These pulses disrupt the cell membrane, enable the efficient uptake of a genetic materials. Electroporation is known for its high transfection efficiency and adaptability to various cell types. Another method is microinjection, where in this technique used directly introduce genetic material such as a DNA, RNA into individual cells. It involves the use of fine glass needle or micropipette to manually inject the genetic materials into the cytoplasm or nucleus of a target cell. The process involves microinjection followed by preparation of the injection materials, preparation of a microinjection setup, third cell preparation, the fourth one then do microinjection, the fifth is followed by validation and analysis. The cell can be further analyzed confirm. Microinjection offers several advantages including the ability to precisely target individual cells, control over the amount of location of the injected materials and suitability for diverse cell type. However, it is labor intensive technique that requires specialized equipped and skilled operators. It is often used in research setting where high precision and control over the delivered genetic material are crucial. Another method is a particle bombardment. In this method, it is also known as a gene gun method, a physically transfection technique used to deliver genetic material such as a DNA, RNA into the cells. It involves the acceleration of bombardment of tiny DNA coated metal particles into target cells using high pressure gas or explosion. In this method, using basic steps like coating of the particle, loading up the particles, then shooting up the particles, which is followed by particle cell interactions, cellular response and analysis. The particle bombardment offers several advantages and including the ability to deliver genetic material directly into the nucleus, the potential of efficient transfection of a wide range of cell types, and the ability of transfect difficult to transfect cells. However, it may lead to cell damage, reduced cell viability due to high velocity of particle impacting the cells. Careful optimization of parameters such as particle size, acceleration force, target cell positioning is necessary to optimize cell damage. The next method is known as a sonoporation, that uses ultrasound waves to induce transient pores into cell membrane, allowing the entry of genetic material into the cells. It involves application of ultrasound waves at a specific frequency, the intensity to achieve membrane permeabilization. Sonoporation works on the basis of different steps like acoustic, microbubbles formation, ultrasound exposure, cavitation events, membrane permeabilization, genetic material uptake, cellular response and analysis. So, sonoporation offers several benefits including its non-invasive nature, broad applicability to various cell type tissues and potential for target delivery in specific areas. It also provides a mean to achieve high transfection efficiency. However, it should be noted that optimization of ultrasound parameter, appropriate selection of microbubbles and consider of potential cellular damage are important factors to ensure successful and safe sonoporation. Now, another example is magnetofection. In this technique used to deliver nucleic acid into cell by combining a standard transfection method with magnetic forces. This method involves use of magnetic nanoparticles that are functionalized with the nucleic acid of interest. The steps in all these processes are preparation of magnetic nanoparticle, functionalization of magnetic nanoparticles, application of magnetofections, then accumulation of magnetic nanoparticles, then internalization of nucleic acid, removal of magnetic nanoparticle from the cell. So, magnetofection offers several advantages over traditional transfection method. It can increase the efficiency of nucleic acid delivery, reduce toxicity associated with high concentration of transfection reagent and allow the targeted delivery of specific cell population by controlling the position of a magnet. However, the method require careful optimization. So, with this discussion, I thank everyone for your kind attention. Hope this lecture has been very fruitful for you. In the next lecture session, we will be able to learn about genetic manipulation in plant in details. We will meet you in the next lecture session very soon. Till then, take care. Thank you.